Welcome to the Nuformix investor webinar. My name's Tim Metcalf, Managing Director of IFC Advisory, Nuformix's financial PR and investor relations agency. The format of this afternoon is Dan is going to run you through a short presentation, providing a brief introduction for those who are new to the company, and more importantly, for those who are familiar with the story, an update on where we are and things that have been recently announced. Following the presentation, we'll have the opportunity for a question and answer session. Really appreciate those who sent questions in advance. They've all been considered and have helped shape in certain aspects what Dan's going to be saying. So you'll see some of them are already covered. Those that aren't covered, I will endeavour to ask Dan at the end of the presentation. Obviously, we would welcome further questions. Given the number of people who are online today, it's not possible to go to you individually, ask you to unmute and put your questions directly. So please do use the Q&A button at the bottom of your screens, type in any questions that you've got, and I will endeavour to ask those to Dan at the end of the presentation. Okay, well, without further ado, Dan, I'll hand over to you to run through the presentation.
Excellent. Thank you very much, Tim. So conscious it's a very nice evening. Conscious also that I imagine quite a few people attending have heard the Nufomix story before. So the slides I'm going to go through, some of them have been presented before. The deck will be made available after the session today. And so I'm going to spend a bit more time on the slide where we've got some new data. I thought it would be really nice to sort of share some of that with you, and maybe go through a bit more briskly the things that we've spent a bit more time on in the past. So as Tim said, without further ado, let's get started. So first of all, just a piece on the team. I'm really proud to be part of the team that is Nufomix. The board is made up of myself plus Maddie Kennedy, who has spent a career in life science CFO roles with a really tremendous track record in terms of M&A activity, and those kind of skills and experience I think are going to become hopefully more relevant given the stage at which the company is at right now in terms of having a lot of data on key programs and really upping the ante in terms of its out licensing activity. Maddie and myself are joined by Julian Gilbert. Julian's also a career spent in biotech companies, entrepreneurial type stuff, very successful exits. Most importantly, I think, within his time at Arrakis, Julian ended up out-licensing an asset that was redeveloped from an oral compound to an inhaled treatment and exiting very successfully that asset in a deal with Novartis. So that skill set, I think, is really important given one of Nufomix's lead programs, which is almost a parallel of that, which is our NXP2 program, which I'll speak more about. Myself, co-founded Nufomix in the past, have been involved in technology business in and around the Cambridge areas, focused on biotech my whole career. For those that don't know me, focused in deal making, fundraising, and these kinds of activities, and transitioning businesses from technology development to product development. In terms of the operational model of the team, we have a virtual model, which is very common for small biotech companies developing a small number of assets. We're supported by therapeutic, which is a company that spoke very well recently about product areas that are very close to Nufomix in terms of inhalation therapies for IPF. The team there are all big pharma experts in terms of their specific therapeutic areas. They help us cost effectively develop our assets. They help open doors to partner companies. We're really lucky to have these guys on board supporting us. And we have a host of other specialists in other areas, IP, etc., to support what the business does. So virtual development team, very low cost way of progressing assets towards key inflection points. So in terms of what Nufomix does, it's focused on drug reprofiling. So that's redeveloping known drugs. And we're able to do that as we have new patents that have allowed us to solve historic problems in the development of known drugs. These patents pertain to improved version of existing drugs, where those improvements unlock a historic problem that's prevented development. We have a program in IPF, or idiopathic pulmonary fibrosis, or lung fibrosis, as we may have heard of. It's certainly something that's very much in the news more regularly as a result of COVID and long COVID. So this program is about a proprietary form of tranelast, and we are taking that program forward as an inhaled therapy, which is a relatively innovative way of treating that disease in terms of IPF. And then NXP4 is our second program in oncology. That's targeting an improved version of Lynparza, which is based on the drug olaparib, which is a PARP inhibitor, a very modern sort of foundation, if you like, of the treatment of cancer. So in terms of how we build value into Nuformix, we have, as I've mentioned, this virtual product development structure supported by external net experts with their own knowledge about our programs. Very lean operating model. We are charged with taking these two programs through value inflecting moments in terms of generating data that de-risks the development of those products further. So we're looking to take these products on to a point of out licensing. Look to build value into the business through those kinds of transactions, be it through milestone payments or future royalties. So that's effectively what the business model is about, focused on these two assets and moving them into an out licensing moment. How we do it? Well, as I've mentioned already, we look to solve problems historically for really attractive drugs. We're not looking at trying to create sort of incremental benefits, make things 10%, 5% better, really trying to focus on some major problem areas that open up new uses for drugs that weren't previously possible. We file patents in those areas. We validate the use of those patented inventions in ways that demonstrate what our products and development can go on and achieve. The redevelopment of known drugs is not a new thing. It's a particularly attractive thing, as people that are, I guess, are familiar with the sort of drug development sector will know that it's a relatively high risk industry. Attrition rates are relatively high in the industry, such that we have something like a failure rate of about 19 out of 20 molecules that go into human clinical testing actually don't make it through. To be able to work on existing drugs changes that risk profile dramatically. We already understand pieces about safety. We already understand pieces about what the drugs might do in humans relevant to the disease problem that we're facing. Also, known drugs allow us to get new therapies where we can get those clinically approved into patients a lot faster. So some real attractions around working in this space in that way. It's also something that's known to create value. Some really nice examples here for those that aren't familiar with them. Biogen, which is one of the main sort of stalwarts that you have heard of in terms of its therapies and things for Alzheimer's recently. One of their major blockbusters until recently was dimethyl fumarate, which was actually known since the late 1800s and was previously a treatment for curing leather. Okay, this is a real bio-focused company. Their greatest sort of sales were achieved from a very old molecule that was developed for the treatment of MS. This concept of molecules that have maybe been discarded or been forgotten about is something that you see a lot of in this sector. A really good example is that of Prevention and their product Tzield, which was actually acquired. They actually acquired the license to this product back in 2018 for 6.1 million. And they were acquired by Sanofi in 2023 for something just short of $3 billion. So these kinds of things where we're taking a known drug, redeveloping it, adding something to it, able to do that in ways that weren't previously possible, commonplace in the industry and shown to generate value. So our focus in terms of NXP2, then switching onto that, is as an inhaled therapy for the treatment of lung fibrosis. Okay, or as it's more specifically known, IPF. IPF is a high mortality rare disease. It's caused by a scarring of the lung, and what takes place is you lose the ability to get oxygen from the air into your lungs and into your blood. It's an orphan indication, whereas it's typically associated with older males. And can be associated with things like smoking or working in more high-risk, say, industrial environments, global prevalence is increasing. We'll also see, I think, a quite significant effect in terms of COVID on the sort of prevalence, I think, of IPF. I've seen studies that suggest that the number of people that have had a COVID quite seriously and have lung fibrosis is anything between 11 to 50%. So there's clearly going to be some factors in this disease and this problem staying around a bit longer. And it is a problem because the only approved treatments that are out there are not incredibly efficacious. They're not massively life extending, and they have a really significant discontinuation rate. The treatment side effect profile is really quite considerable and cause a large majority of patients to discontinue. And that's in spite of actually not showing much improvement in their symptoms. Very high cost healthcare burden as a rare disease. Typically, treatments are very expensive. Lung transplants, as we'll talk about, are also one option for sort of improved treatment, but they're certainly not something that can be used with a large number of patients in this space. So really the outlook for this and being diagnosed with this is not great. Poor quality of life and reasonable sort of mortality. So our concept then, in order to sort of do something about this and improve the sort of treatment of this disease, was to focus our therapy around a molecule called tranelast, which was actually first invented by a Japanese company called Kisai. It was approved across Asia for skin scarring, which has relevance towards scarring of the lung and the loss of the function in that tissue, and asthma, specifically related to inflammation. So fibrosis, inflammation, which turn out to be two really important components of IPF. Kisai didn't opt to develop the drug elsewhere. Therefore, in territories such as US, which is very important towards the sort of global IPF market, the drug will be treated as a new chemical entity there. So in short, excellent history in terms of relevance towards the sort of disease mechanisms that play into IPF. So the issue that we had was that oral delivery, as the drug is currently approved for, is not something that's going to be suitable for chronic diseases, particularly where we need to get a high dose of drug on board, as is the case in IPF. So what we've opted to do is look at inhaled delivery via nebulization. This allows us to direct the drug right to where it's needed, into the deep lung, and this offers a number of benefits: avoidance of oral delivery challenges, avoidance of side effects. And we're now in a position where key patents have been granted in territories such as the US and Europe that give us long-term protection of this invention. Our ability to deliver tranelast via inhalation is at the heart of the NXP2 program. And a little bit about this, the people have seen this slide before, and we'll make it available. This tells you a little bit about what happens in terms of how IPF comes to pass. Usually there's there's two components which I will focus on for good reasons. I'll come back to some important advances that we've made in the data. We have a stimulus. This could be things that people might be familiar with, maybe in terms of smoking or asbestosis or something like that, where there's been a lung injury event, a tissue injury, and that's kicked off a progressive fibrotic process. And that fibrotic process doesn't actually stop. It just keeps on going very slowly in the background. You have inflammation, which is driving this kind of excessive wound healing, and you get this kind of buildup of collagen and excess tissue, and that functional tissue within the lung that's doing all the essential jobs of getting oxygen into the blood is unable to do that anymore. And eventually, we just simply are not able to get enough oxygen on board to survive. So we have a fibrotic piece, we have an inflammation piece, and it's really important to get this across. As I mentioned, COVID will have an impact on the number of patients suffering from this disease, and as will, I believe, changes in quality of air, etc. So these kinds of problems in relation to this disease are here to stay. So, as I've mentioned already, what's our solution in terms of NXP2? Why is our approach so attractive? So, firstly attractive. We've identified that tranlast is a really attractive drug for this disease, got great mechanistic evidence there. But as previous studies have shown, that oral dose is a challenge in terms of long-term tolerability, in terms of the ability to deliver the drug successfully. These are very difficult things to do with this drug. And then one would automatically switch to thinking about inhalation. However, inhalation is really challenging for this drug as it is as well. The drug is incredibly insoluble, which means that dry powder formulations are simply cleared from the lung. Nebulization as a solution is not an option because you can't actually dissolve the drug in order to be able to nebulize it. Turning then to thinking about inhalation as a way of treating IPF, this is a really interesting opportunity to solve some of the issues that patients have in use of existing therapies. Existing therapies are used orally, which means that they have, as well as the sort of on-target effects of treating disease, they have a lot of off-target effects, and that's the reason why we see such levels of discontinuation. So the idea of delivering the drug direct to the lung, maximizing it where it's needed. In order to be able to do that successfully in IPF, we need to be able to get the drug to the deep lung, because that's where fibrosis is taking place, and that can only be done with nebulization. Also, IPF patients, they don't have great lung function, so they aren't able to use inhalers. Therefore, the concept around being successful in this space is with a nebulized approach. We can get to the deep lung for IPF patients where we need to. They don't need a great lung function to be able to use a nebulizer, but we need to be able to make the drug soluble. And that's exactly what we've done with our patented drug form, NXP2. So we now know that we can dissolve this drug in a solution suitable for nebulization. We can therefore unlock the ability to get this drug into the lung and use its pharmacology to treat this disease. The drug's clinical safety profile is really well understood. And so we really believe that there's great scope for a kind of a therapeutic index, if you like, via inhalation, seeing good efficacy, but without the downside of side effects. Another reason why this is an attractive thing to do, other than solving the lack of effective therapy, if you like, is the commercial opportunity. As rare disease therapies, treatments per patient per year can cost in excess of hundreds of thousands of pounds, and these end up being quite large markets. So these are sort of multi-billion-dollar markets projected to reach something like £8 billion by sort of 2030. The existing therapies that are there, if you look at Ofev, which achieved its greatest sales on record in 2022, something like £2.8 billion. Esbriet coming up behind it, that drug became off patent in 2022, and therefore its revenues have started to reduce there, but that was peaking in terms of peak sales as well at well over £1 billion. So these are really attractive markets to be in. And then also, as the next, I guess, connection as to where we come in and what our plan is to look at out-licensing NXP2 to a development partner. When one looks at the kinds of deals that are done and their stage of development, be it preclinical, phase 1, phase 2, and phase 3, we've looked at deals that have been done in the space here, and averaging out those deal values, you can see that there are some quite attractive deals to be done should one be successful at out-licensing assets at either of these stages. We'll be looking at out-licensing or partnering NXP2 at a preclinical development stage currently. So some new slides here then that help perhaps explain the sort of approach that we've been taking when we've been trying to build out this sort of supporting data set to help us go off and complete one of these licensing transactions. So first, starting off on the left-hand side and moving towards the right, we need to build out an evidence base that tells us that essentially NXP2 can be commercialized successfully as an inhalation therapy for IPF. And in order to be able to achieve that, there's a number of things that we need to be able to demonstrate at a preclinical level. First of all, is efficacy towards fibrosis and inflammation. Those following the story, they've seen that we've made various announcements about that, and tranlast's ability to sort of demonstrate this capability is well validated in many models. We need to demonstrate that we can use our soluble form of tranlast to create nebulizable solutions that get the drug to the deep lung. we've been able to do this, and we've got some nice data that we can show you shortly in terms of showing what happens when we're able to do that. And consequently, from doing that, we need to see that the drug thirdly then exerts its pharmacology following inhalation. As people that follow the story have known that we've done that. All of these boxes are now progressively getting ticked. More recently, what we've demonstrated is that the drug has a sufficient duration of action. Anyone who happens to be using an inhaler for treatment of any kind of condition will know that ideally one would take an inhaler relatively infrequently, perhaps once or twice a day. In order for that to be useful as a therapy, following inhalation, the drug needs to have a long duration of action. It's not going to be practical for a patient to be using an inhaler 4, 5, 6, 7 times a day in order to get the drug on board and keep it there in order that it can treat disease. So we need to be able to demonstrate that we've got duration of action. Also, as is becoming absolutely essential for clinical development and commercialization, we need to demonstrate that the drug is compatible with use of standards of care. So the standards of care are the approved therapies in IPF, which are nintedanib and pirfenidone. We need to demonstrate that we're able to add efficacy on top of those treatments and perhaps offer the potential to reduce side effects as well. And we've got some pretty compelling data supporting that as well. Lastly, the drug needs to be tolerable in the lung. Well, what we've seen so far is that we've explored in a preclinical setting, delivering very large quantities of this drug to the lungs of preclinical species, and we've not seen any kind of tolerability or toxicity programs. So going forward, as we start to build out our evidence base here, there were 2 attractive opportunities for us. That is to increase the efficacy of existing therapies and get a better handle on disease progression and extend life. We have to do that in partnership with existing therapies, or potentially as a standalone therapy for those patients that are declining use of standard of care treatment. Moving on then to some of the data that supports these 6 kind of pillars, if you like, of evidence base that we're trying to build in order to support a sort of out-licensing event. People have seen some of this data before, but this is a preclinical model of the disease. It's something called a bleomycin model, and this model simulates an inflammation event. And one of the sort of major challenges that drug companies face in this particular disease areas is that the models for investigating whether a drug has got potential to treat a disease are really, really challenging. When, as I spoke about earlier, you get this event that is responsible for the start of lung fibrosis. This can then be something that the difference in time between that event and when a patient presents himself at a doctor's short of breath or whatever can be something like 20 years. It's a really difficult process to simulate within a preclinical species, for example. So this preclinical model here, we're looking at where we have both a treatment with bleomycin, the BLM, and we have a treatment with bleomycin and tranilast. And we can see from the column second from the right, this is measuring the amount of fibrosis in the tissue. And what we're seeing is that when we follow treatment of the sort of bleomycin, which causes the fibrosis event to occur, and we follow that up with tranilast, we can reduce the amount of fibrosis that we're seeing in that score. So really interesting story about this, speaking to potential partners. Some partners absolutely insist on seeing an oral bleomycin model, but they actually don't seem to mind too much whether the molecule fails or succeeds in this study. So it can tell you that actually, which I find really surprising, the weight given to some of these models, it's a really tricky space to be in. That's why it's really nice that we're able to point to some of the findings of those by academics in the public domain that have published some of their work following the COVID pandemic, looking at patients that have suffered with severe fibrosis following a really poor COVID infection. So we've seen patients treated in a number of different examples where, thankfully, due to certain geographies where tranilast is approved, physicians have made the connection between the drug and made the connection between what it can do in terms of an IPF setting and looked at dosing to patients for a short period to look at how tranelast ameliorated their sort of lung fibrosis. And two excellent examples here of where the drug was really successful in doing that. Of course, short periods, as I've mentioned, of treatment are not going to be viable. We need to look at long-term treatment for patients with chronic disease. So this is really useful research in terms of validating the concept, but does mean that it's inhalation that we really need to focus on in terms of getting this molecule into these kinds of treatment areas. So another area of research that we've been focused on that maybe takes away from some of that are used more traditionally in the space, we've actually focused on human IPF tissue. So as I mentioned, some patients are lucky enough to receive a lung transplant. If they get to end of life stage, they're on a transplant list, a suitable pair of lungs comes up and they're able to receive a transplant. We've been working with a company that's been able to take the tissue and use that tissue as a testing ground to look at the efficacy of potential therapies for IPF. And what we've seen is we've put our drug, NXP2, up alongside pirfenidone, which is one of the approved therapies. And what we see is that this gives us an idea about the amount of collagen that's being expressed in terms of the connection with collagen, and that has increased levels of fibrosis. What we're looking at here is the ability to reduce the expression of collagen 1A1 over a time period, so up to sort of 96 hours, and we look at each day over that kind of a period. And we can see we get to this amount, sort of 100 micromolar sort of dosage, and we're not really improving NXP2's performance here. We've reached the maximum, which is great. And when we compare how we're doing to pirfenidone, we're doing a lot better in terms of reducing the expression of collagen, but we're also working at something like 25 times lower doses. So this is the sort of first indication we're working in a really high challenge disease model, something that's also super close to patient, and we seem to be performing incredibly well, which is really exciting. And this is one of the sort of pillars that I was talking about earlier in terms of building the story around the molecule and its use in IPF. So that's all well and good, and it tallies well with what's known in the public domain. This concept of switching tranelast delivery from oral to inhaled, and the ability to exert the drug's pharmacology in that kind of way is then the next piece that we need to be able to validate. And that's exactly what we've done in terms of a dose response study. So on the left here, what we're looking at is the expression of two biomarkers of the progression of fibrosis, one in relation to fibrotic pieces, and the second in relation to inflammation. And what we see is that as we increase the dose in these kind of circled areas here of NXP2, we see a dose response. We see a reduced expression of these kinds of biomarkers in tissue. And so this is, first of all, validating that the molecule can exert its pharmacology following inhalation. The next piece, which is something that we've spoken a lot about, which wasn't established until very recently, was the molecule's duration of action following inhalation. As I mentioned, NXP2 would struggle to be a viable therapy if patients were having to use it 4 or 5 times a day. So just to sort of show some data here, what we're looking at is human healthy lung tissue. Okay, this is challenged with a stimulant. The tissue is allowed to sit in drug for a short period, and then it's challenged at various different time points following treatment with drug to see how long the drug hangs around for and how long it can exert its pharmacology for. So here on this first 2 columns on the left of this graph and the duration of action, we can see what happens in healthy tissue when it's challenged with LPS. We see this spike at MCP1. Following treatment with NXP2, an hour later, what we can see is we treat with LPS again, and we see this much reduced expression of MCP1, telling us that the drug is doing what we expect it to do. If we now look at the sort of 12-hour picture in the control here, what we still see is that even after 12 hours of just having exposed tissue to drug, we still see a marked change in the amount of MCP1 that's being expressed, which is really encouraging. This tells us exactly the kinds of messages that we need to have in terms of addressing the objections that potential licensee partners are going to have. We can demonstrate that the molecule can exert its pharmacology following inhalation. And we now know that NXP2 has got a suitable duration of action for twice daily dosing, which I think is going to be the right kind of product profile in clinic. Going further then, you've also heard about the importance of understanding how NXP2 works with standard of care. Any patient, well, the vast majority of IPF patients diagnosed are going to be on either pirfenidone or nintedanib as treatment. They may well be on reduced doses of these drugs in order to manage side effects, but if patients are already on therapy, we need to understand how NXP2 works alongside those existing therapies in order for clinical research to be done. By turning to standard of care type designs in terms of clinical research, we know we can recruit patients quickly and conduct clinical research towards approval much more rapidly than if we were looking for naive patients that haven't received either therapy. So really, this is a really important thing to be able to understand. So just generalizing very broadly then, we're looking at how NXP2 adds performance to standard of care in terms of fibrosis, but also in terms of inflammation. And to generalize a little bit, what one would probably say is that pirfenidone is reasonable in terms of its anti-inflammatory activity, but not so good in terms of its anti-fibrotic activity. Again, here we're looking at the amount of collagen 1A1 expressed. We can see that in vehicle, we see a very large amount of collagen 1A1 expressed. And this is also, again, a study that's being done in human IPF tissue. When we treat with NXP2, we see a significant reduction in that. When we treat with pirfenidone, we also see a reasonable reduction in that, even though it's being used at something like 20 times the levels of NXP2 required. When we add these two together, we see a really synergistic effect. We see a real lowering of the expression of collagen 1A1, which tells us that these two drugs combined can have an increased ability to treat the fibrotic aspect of IPF. If we switch over to nintedanib, nintedanib is quite a reasonable anti-fibrotic agent, but it does struggle a little bit in terms of its inflammation. As it turns out, in the study that we ran here, actually nintedanib had quite an unusually strong effect in terms of anti-inflammation, which happens in sort of, I guess, about 25% of cases. But here again, what we see is that we see a really strong anti-inflammatory effect versus MCP1 for NXP2. We can see that those levels aren't quite achieved by nintedanib, but certainly when we add tranilast and NXP2 into that equation, we see a much better response in terms of the expression of MCP1 and what that correlates with in terms of improved treatment of the inflammation component of IPF. So that's really the sort of package that we've been looking to build, to be able to have preclinical data that supports each one of these pillars that we've identified, but also to have it in studies that validate each other. For example, just coming back to these two data points, in the sort of preclinical inhalation dose response here, we've used an LPS challenge. In the human healthy tissue here, we've also used an LPS challenge, which is a really nice story to be able to tell in terms of the translation of what we're seeing in terms of how the drug works from preclinical into a more close to patient type model. So just switching away then to from NXP2 to NXP4, speak a little bit about that program. As we've spoken about before, Lynparza is now a mainstay of modern oncology therapy. It was the first PARP inhibitor to reach approval and achieves considerable sales for AstraZeneca and is patented out to sort of 2029 in terms of its formulation IP, in terms of how that extends its patent protection. Our goal here is to look to potentially partner what we have in this area with either the originator, AstraZeneca, in terms of a sort of an opportunity to make a better product and allow those companies to lifecycle manage their products, or to partner with another third party, either a generic or somebody that's very interested in getting into that kind of product space. The formulation as is, the marketed formulation of Lynparza has seen some changes over time because it's a very challenging drug to deliver consistently. What happened initially was that it was originally marketed as 8 50 milligram capsules given twice a day. AstraZeneca was able to modify that situation and reduce a second product onto the market, which ended up having a sort of a significant increase in bioavailability, and therefore a reduction in the sort of pill burden that patients have to take. So we've actually looked at comparing and contrasting how our co-crystal based formulations of NXP4 perform against the marketed formulation of Lynparza, that tablet seen on the right hand side, in key dissolution studies. Dissolution studies for this molecule give us a good indication of how we may be able to improve the absorption of this drug in the clinical setting. So this is looking at dissolution data. We've presented this data before. On the left-hand side, we have the dissolution of the marketed product. And on the right-hand side, we have our co-crystal. And effectively, what we can see over time is that we have a significant increase in the amount of free drug dissolved in this particular study. And the expectation is that this will lead to enhanced products with greater bioavailability. We were looking to do that as a next step in terms of validating that in a suitable preclinical pharmacokinetic model to support what we're looking to do in terms of licensing out this asset. So I hope that's given you a bit of insight into Nufomix if you've not heard about the business before. I hope that gives you a good coverage of our activities. For those familiar with the story and keen to see some of the new data, I hope that's been useful for you in terms of understanding why it's been important for us to build out the data sets that we've targeted, why it's been important for us to understand how NXP2 works alongside standard of care and the extent to which it improves those kinds of therapies. Also, why we've been so keen to demonstrate that there's a suitable duration of action and that we now seem to have established that, and what that means in terms of the completeness of the data that we've got to now go off and have those kinds of next stage partnering discussions. So just summarizing then, and as a close to sort of look at the outlook for 2023 and beyond. So we will look to continue this virtual development model that we have and lean operating model going forwards. We've just got some very specific investment that we're looking to do into R&D with NXP2. We are now in a position where we don't have to do any new work that we haven't yet completed. We're simply looking at increasing the sample size of data sets that we have so that we can make those data packages suitable for partners in the data and the statistics. In NXP4, sorry, as I've just mentioned, we're looking at in vivo proof of enhanced bioavailability. And really what you're seeing now in terms of the business is a shift in focus from building out these data packages that support out licensing to securing development partners in these key programs. And that will be what the remainder of our activity focuses on for 2023 and beyond.
Well, thank you very much, Dan. Excellent presentation as always. Really appreciate it. Thank you very much for those who've been typing in questions. And as I said at the beginning, thank you for those who've submitted them in advance. Before I start running through the questions, I must say that unfortunately one or two that have come through are touching on subjects that no public company can comment on or answer, specifically around giving forecasts as to financial numbers, what may come in, and timescales for future announcements. But we will do our best to go through things. I've tried, while Dan's been speaking, to group the questions into a number of areas, but excuse me, do excuse me if I dot around a little bit. But Dan, starting at the most obvious point with NXP002, just a few clarifications. You've talked about licensing partners discussions. Can you confirm that those are underway and there is genuine interest out there, and you do believe that this is a program that's worth continuing? One questioner asked.
Yeah, absolutely. We started, I guess, formally this activity in terms of putting NXP2 above the parapet at the European Respiratory Congress last year. And started initiating contact with companies then. That was not the first time, obviously, that the business had specific out-licensing discussions with respect to the program. As I mentioned, we've built a data package that is designed to address specific objections that we've had from existing previous discussions. So the kind of activity that we've been undertaking is to look at those companies that we've had previous contact with, where we have existing relationships with, and just reopen the door and go back and share some of this new data that supports some of the questions and outstanding issues that were previously raised. So that activity is underway. The other kind of activity that we're looking at are just understanding how those companies that are seeing the global IPF market as being attractive are looking at dipping into that market potentially, who those companies are, and just building out a target list of companies that we want to take the NXP story to. And that's a more specific longer term activity, building out the story, approaching companies with the right kind of message and establishing contacts. That's activities underway as well. The European Respiratory Congress meets this year in September in Milan. We hope to have a considerable number of discussions at that kind of event, with obviously discussions already underway with a discrete number of specific partners.
Excellent, thank you. I've had a number of questions surrounding, were you to do an out licensing deal, what would be the next steps for a partner once you've taken it from sort of Nuformix stage to involving a partner?
I think it's worth saying as well, maybe just one point that in order to answer this question, it's going to be quite interesting to see what form a partnership takes, and it could be anything from out-licensing the asset as a whole, where simply hand the asset over to the partner and they look at developing it themselves with a team, or I guess another slightly different version of that would be full outright M&A, acquiring Nufomix lock and stock and all of its programs, or the third opportunity would be something that would be more like sort of collaborative development, where a company may secure an option or a right of the program, and the two companies, Nufomix and the partner, would work together up through those next stages. So the next stages would be that we would need to secure preclinical data around tolerability in the lung. So that would be a box that would need to be ticked before we can get into sort of human studies, clinical studies. However, in terms of looking at that data, we've done a lot of the precursor work already. We've seen no sort of issues in terms of any sort of tolerability, sort of red flags or anything like that. So that's simply a data package that needs to be acquired in order to move into sort of human studies. The next stage from that would be looking at studies in healthy volunteers, and that would be dosing the drug to the lung to look at concentrations of drug that are achieved both in the blood, but also in fluids in the lung, and just trying to understand, what kinds of doses are we given and what's that achieving in terms of exposure. And once that picture then begins to build, one could take a view based on some of the preclinical data on what kind of exposures that we need to achieve in order to have efficacy. From that, one can start to understand what doses one would look to want to investigate in a patient study. And so the next stage from that would be going into what's called a phase 2A study, which is your first venture into patients. You might look at something like a 60 to 80 patient study over something like 12 to 24 weeks initially. There's a lot of advancement that's been made in sort of imaging technologies and other sort of biomarker related aspects that help us understand disease progression a lot more quickly now in this disease space. And so from that, assuming success, that study might transition into a phase 2B study, be just studying that patient group for a longer period before then going into a phase 3, which is your market authorizing study, and then achieving approval. So they're the kind of steps, if you like, that any partner would need to achieve in order to actually get the product registered. There would be shortcuts potentially around that if one was to work in areas where the drug was already approved. There would be shortcuts if one was able to focus on specific patient populations in rare diseases or rarer forms of lung fibrosis. But hopefully that paints the full picture in terms of what a standard process would look like and what some of these opportunities are for speeding that up.
Yeah, thank you. Now you talk about the final step, and we understand that Tranilast is only registered in Japan, or licensed in Japan and certain other Asia Pacific countries. Does that actually provide a challenge to bring it to NXP002 to other markets where Tranilast isn't licensed at the moment?
I think the challenge, the word isn't challenge. It just means that there's just got a couple of extra steps. I think, you know, the sort of the path towards generating data to support entry to clinic is really well trodden. You know, it's not a challenge in that it's difficult to do or risky to do based on the data that we've got. It's just something that regulators will need to see. They'll need to see a safety data package that supports the doses that you'd like to give into humans. So yes, you know, one would like to think that if there were other geographies that where the drug was approved, you might be able to get sort of faster into these kinds of informing studies. I would like to think that that there wouldn't be much of a difference in terms of conducting some of the early stage clinical work in geographies where Tranilast was already approved, and how that would inform dosage that you would select for a patient anywhere in the world. And so, you know, that might be one, you know, really kind of a attractive approach. The other benefit, though, of course, is that, you know, you talk about challenges. There's also benefits in that if you were to look at the US and Europe, which are the main markets for IPF, Tranilast would be considered a new chemical entity, and would therefore benefit from longer sort of periods of exclusivity and protection for anyone developing it and taking it into that sort of territory as well. So, which is attractive in terms of those regulatory benefits.
I've had a number of further questions. People are obviously very interested about the potential use here. You mentioned IPF is a rare disease. Should we get to the stage where 002 is generally available, is it something that's likely to be provided by a clinician under clinical supervision in a hospital, or is it something potentially that could be much more widely used?
Oh, I think in terms of wider use, I think it'll only be used for the patients in which it's diagnosed for as part of a clinical study, and at the moment, IPF is a rare disease. It's actually one of the larger rare diseases, though, in terms of how orphan designation is calculated. It is one of the more prevalent sort of rare diseases. So the ideal way in which I envisage the product getting approved is as a sort of a for use alongside a handheld nebulization device. This would be the kind of thing that a patient would use at home or wherever they were, fully sort of portable and usable anywhere. So treatment would be within the home. One of the things that has come up with Tranilast is that it does have a really attractive sort of anti-inflammatory profile. Some of the relevant additional conditions that one might consider are things like acute respiratory distress syndrome, which was something that was associated with problems in relating to COVID. So that's also a potential area of pivot into that space, or a different area where you might see Tranilast used in future, depending on the sort of development of direction. But the real core would be seeing it used as a nebulizer by patients that had already been formally diagnosed with IPF or related fibrotic lung condition, and that's the main thrust of where we see things going.
Just a clarification question that's just come up, moving back to the work that Nuformix is doing. It did take a little bit of time to get donor lung tissue for the last studies. Do you feel that there's going to be any sort of impediments to further work for getting tissue samples?
Yeah, you're right. It was there was a period where we did struggle. Actually, it just so happened that everyone that was at the top of a transplant list was seen to be a COPD patient or something else, and we did for a little while struggle to get the right kind of tissue. However, I think we were also the first company to access this new version of the model, which uses much larger numbers of repeat tissue samples and generates much more consistent data. This is a model which is highly attractive to all drug companies looking at developing drugs in this space. It potentially replaces as the gold standard for all kinds of preclinical R&D in this space. and our partners, Fibrofind, or obviously would like to make that model widely available to as many partners as possible. And therefore, what they've been able to do is expand the relationships that they have with treatment centres performing lung transplants. I believe they've expanded that now to Papworth here in Cambridge, which is the second largest lung transplant centre. So they have now got a vastly increased access to the right kinds of tissue, which should mean that should we, as we look to just increase the statistics and the repeat numbers in terms of the work that we've done, that isn't going to be a problem.
Excellent, good to hear. I think I've covered all the 002 questions. Moving on to 004. Really just a clarification, are the programs here progressing, or is it on the back burner subject to funding and all the focus on 002 at the moment?
No, I think we understand really well exactly the kind of data that we need to generate now, and I think that's something that we've been focusing on. What kind of data is going to be most supportive of demonstrating a benefit and advantage of the product, and we're planning on conducting those studies in the coming months. It's not a particularly expensive study, and we hope to be able to progress that asset. It's the reality is it's not the value driver, I guess, the main value driver behind Nuformix. NXP2, I think, has clearly got the potential to transform a really tough to treat disease, and therefore that's why we spend a bit more time talking about it. But that doesn't sort of diminish in terms of NXP4 and the opportunity that we have there. Also, it's in some ways that the kind of pressure's not on in terms of that asset. If you look at the sort of timelines that I spoke to in the slides, you look at patent expiry in this space is something out till sort of 2027. So generics looking to get into that space, maybe even sort of 2029, can't do so for still quite a long time. It does give us an opportunity to build the data sets that we need to be able to engage with the right kind of partners in the right kind of way. So maybe another reason why NXP4 isn't spoken as much as well is because there's that window of opportunity in terms of being able to commercialize that asset.
Okay, thank you. And moving on to 001. Anything more you can say about progress since the out-licensing, and also talk a little bit more about the relationship with Auxilio and how that works day to day?
Yeah, of course. So previously, Nufomix took NXP001 into the clinic. So got a really good understanding of what's required to use that novel and proprietary drug substance to make improved versions of that drug. Auxilio have licensed that program in its entirety and are now responsible for making all of the development decisions about what happens with that asset. That's not something that Nufomix is involved with. That's entirely driven by Auxilio and their decision making as to what they develop and how and when. Certainly, Nufomix is on hand to support that, in detail. We give regular updates as we receive them from Auxilio in terms of what their activities are. They made a lot of progress. They're looking at doing something very different. Auxilio, it's looking at developing a new treatment for cancer itself, not the actual side effects of cancer treatment, which is what the molecule they're concerned with, the Prepsin, is approved for. And so that requires some very different thinking. I've been really impressed with what I've seen recently from what Auxilio have been doing, and we'll continue to update shareholders and the market in terms of events specific to Auxilio, and hope that we'll have some news on that in the not too distant future. Okay,
so we can assume that there is still potential value there for Nufomix.
Oh yeah, absolutely, 100%. Yeah.
Excellent. Well, moving on, we've got some more sort of general corporate questions, and I have promised a number of people that I will ask the questions as delivered. The first one's about you, Dan. Can you explain a little bit more about your role in the company? The questioner said they thought it was just business development, but you seem to be doing everything now.
Well, it's a small team. I think doing everything. Nuformix is always a small team. In terms of NXP2 and the programs there, that's ended up being, a disease area in which I've been really interested. I think that, a lot of the kind of thinking and the strategy there, I was responsible for a lot of that, making the switch away from an oral therapy into an inhaled therapy. A lot of those, decision moments were things that I felt the data pointed towards us doing. And so, that's probably why, as a program, it's something that I understand. I've made connections with key opinion leaders, development partners, experts. That's a program which I'm really motivated to take forward and understand well. But the reality is that Nufomix is a company developing 2 programs. The requirements to progress those programs, they don't need a full full-time team of different sort of disciplines and things driving those programs forward. And we're at a moment where we really need to control costs. So it makes sense to be able to find a way of progressing these assets in a way that minimizes the kind of personnel and these kinds of other aspects that contribute to the bottom line, and that's what we've been successful in doing. You look at the team mentioned. It's got all the right skills it needs. It's got somebody on a financial level who's wonderful in terms of M&A. Julian Gilbert, as I've mentioned, he knows exactly what to do in kinds of these kinds of situations and moments in terms of getting NXP2 to a point of where we can execute a deal. That's what he's done sort of throughout his career, both in private and listed businesses. So although it might look like maybe me being here and doing all of the talking, I can guarantee you there's a team behind me, supporting me with everything that we're doing, be it a board level, but also in terms of some of the sort of virtual team that I mentioned that are responsible for helping us take some of the programs forward as well.
Right. And that leads me neatly on to the next point that people are making. Question says the company's low cost base is very welcome. But can you comment on how comfortable you are with the current level of funding? Obviously, there is a funding stream coming in from Lansted until October this year, I believe, dependent on the share price, but that's coming in on a monthly basis. But outside of that, are you comfortable that there is the funds to do what you've been talking about today?
Yeah, of course. So no, I can understand where this sort of comes from. It's I think it's no secret that in our industry or any other industry, it's a really challenging time to raise money. What we have focused on is 3 things: maximizing non-dilutive income, and that comes through some of the benefits of R&D tax credit. That is an important income stream for a business like ours. We also, as we've mentioned, NXP1 and whether that has sort of ability to generate non-dilutive income as well. All of these ingredients are there for us to progress forward. I think our funds aren't at a level where I'd love to be able to accelerate everything that we're doing and do this and do that and maybe move ourselves at the moment further up the value chain. So whereas the funds aren't in place for us to sprint, headlong into sort of clinical development, they do allow us to have a really good sort of runway in terms of completing a licensing transaction, and that's what's important.
The next one, again on funding, is probably a completely unfair question, but I'll ask it anyway. Can future fundraising be done by direct share purchases? Can you fund the company, sir?
Yeah, well, I can, I think it'd be unfair to answer on anyone else's behalf, so I'll just answer on my own. I would love to have had a very large exit event in my life. I haven't done that yet. I have a reasonable share in the business, and I'm hoping that I can achieve that via that kind of a route. I will say this though, so I'm not currently myself in a position to fund the company. However, and maybe it's become clear throughout sort of walking through some of the slides and things, if I did win the lottery at the weekend, I would have no problem investing into this program, because I just really believe in it. All the data makes sense. The opportunity is there. So if I could, I would be my answer to that question.
Excellent. Thank you. Just a couple more that I can comment on because they've got a regulatory background to them. The question asks, who was the institutional investor that invested in the last placing and why haven't they disclosed their shareholding? I'm afraid we can't comment on that. It was an institutional investor. They wish to remain confidential for their own reasons. The disclosure regime is actually quite complicated and it depends on the type of institution and how they hold the stock, and whether it's in more than one basket as to whether they've got to file a TR1, but we're comfortable that they don't, and unfortunately they will remain anonymous. Next one says, why are Lanstead continually selling when we were assured previously that they wouldn't be dumping their shares? Well, to the best of our knowledge, they haven't been selling in certainly significant amounts. Looking at my last other screen, 25th of April was the last TR1 that we received that showed that they had 4.82%. Obviously, if they went below 4%, they'd be required to send in another TR1. That hasn't been received by the company. We certainly believe that they comply with those obligations, and should a TR1 be received, it will be announced without any delay. But you know, we don't have any other visibility and can't comment on that one. I've got a couple of questions on takeovers, which as an ex-M&A banker dealing with the takeover panel, I will not put to you, Dan, to save your blushes. But I'm very conscious that we're a couple of minutes past the allotted hour. So the final one I've got on my list here, which I think you've broadly answered, is what's the roadmap from here to a multi-million-dollar company, please? in 10 seconds to sum up.
A multi-million-dollar company. Well, you know, technically we are still worth over 2 million. That is still multi-million. But you know, the route is to secure a development partner, be that through a collaborative development agreement, a single out licensing transaction, you know, whatever it needs to be in terms of seeing the asset developed in the right way. You know, my preference would be that we would find some collaborative development partner, and that a lot of the sort of knowledge that sits within the business, the network that it has in terms of, you know, certainly for NXP2, you know, can go forward and be a part of building value and some retained value in progressing that asset up the value chain. So that would be my preferred path.
Excellent. Well, I think that's a good way to end things. So thank you very much, Dan. But most of all, thank you very much for everybody who's attended this evening. Really appreciated, particularly on a hot evening in England as it is today. Any further questions, please do get in touch, Nuformix at investor-focus.co.uk, and we'll do our best to answer them. But thank you again for your time, and thank you, Dan.
Thanks, everyone.